Ladies and gentlemen, hereby I open this academic ceremony in which Adele Veronika will defend the academic thesis, Origin versus Context, defining key determinants of human macrophage function in health and disease. May I invite you to present a summary of your study and the conclusions of your thesis.
Dear Pro-Rector, dear members of the Corona, dear colleagues, friends and family, welcome to the defense of my PhD thesis entitled Origin versus Context, Defining Key Determinants of Human Macrophage Function in Health and Disease. The disease I have focused on in my PhD research is cardiovascular disease. Cardiovascular disease is the leading cause of death worldwide, accounting for 30% of deaths each year, and the number of people dying from cardiovascular disease is steadily rising. Cardiovascular disease refers to a number of diseases of the heart and blood vessels, including heart attack and stroke. The main underlying cause for cardiovascular disease is atherosclerosis. Atherosclerosis is a chronic inflammatory disease, meaning it develops very slowly and inflammation plays an important role in its development. In a healthy blood vessel, the blood can flow normally. In a blood vessel with atherosclerosis, the vessel is narrowed by a buildup of dead cells and fat, making it harder for the blood to flow, like a blockage in a pipe. If the plaque continues to grow, it can become unstable and break open or rupture, exposing its inside to the blood, making the blood clot. If the blood clot is large, the blood flow will be blocked completely. If this happens in the vessel leading to the heart, this will cause a heart attack. If it happens in a vessel leading to the brain, this will cause a stroke. Thus, while a plaque may go unnoticed when it's still small, it can have detrimental outcomes later on. And in order to prevent atherosclerosis, we need to understand how this disease develops. So let's have a closer look inside the plaque. Inside the plaque, you can find lipids or cholesterol, different types of cells like immune cells, and molecules like proteins that are produced by the cells and in turn have an influence on these cells. The cells I have focused on in my PhD research are macrophages. Macrophages are immune cells that are very good at sensing what is going on around them or in their environment and responding by changing their behavior. In the plaque, macrophages perform several tasks that influence how the disease develops. We call these tasks macrophage functions. In the plaque, macrophages take up particles and lipids in an attempt to clean the plaque from the blood vessel. They produce proteins that can promote or suppress the inflammation going on in the plaque. If macrophages die and cannot be cleaned away, they contribute to the growth of the plaque. Moreover, the shape of a macrophage can tell something about its activation status. For example, a macrophage ready to fight bacteria will look spiky. All of these functions and more we can measure with the macroscreen, a platform based on automated microscopy that I have used in my PhD projects. In the human body, macrophages develop from monocytes, their precursor cells. Monocytes can be found in the blood, but they leave the circulation and develop into macrophages in tissue. In the laboratory, we can use blood from blood donors and take out the monocytes. With good care and the right signals, called growth factors, the monocytes will develop into macrophages, which we can use for our experiments. This process takes 7 days. From this, you might think that the development from monocyte to macrophage is quite straightforward, but actually it looks more like this, a winding road with many turns. You can think of the development from monocyte to macrophage as a marathon. So let's imagine you are a runner. Before you start running, Your fitness level will already partly determine how you will reach the finish line. Are you a trained professional? Do you have the natural physique of a runner? While you're running, are you running with friends who can motivate you? You might reach a crossroad and need to take a decision which direction you will go. It could be a shortcut, but it could also be a detour. And even if the finish line is already in sight, there might still be hurdles that you need to overcome. So now let's translate this to biology. There are actually 3 different types of monocytes, or subsets, which are believed to have different functions. Thus, macrophages coming from different monocyte subsets might also have different functions. In the laboratory, we can culture macrophages together with a low or a high number of other cells, so at a low or a high density. Depending on the density, the macrophage function might be different. Monocytes need growth factors to develop into macrophages. However, the type of growth factor they encounter might influence the macrophage's function. And macrophages are really good at sensing their environment. Thus, environmental challenges, like excess lipids or viruses, impact macrophage function. All in all, this led us to the hypothesis that macrophage function is influenced by their monocytic origin, or the monocyte subset they come from, the density they were cultured at, the growth factors they encounter, and environmental challenges. The aim of my thesis was to use the Macroscreen platform to measure macrophage function during their development from monocytes. In Chapter 3, we isolated monocytes of all 3 subsets from donor blood and let them develop into macrophages. We noticed that at day 1, all 3 monocyte subsets looked like typical monocytes, small and round. However, after 7 days, only one of the subsets looked like a typical macrophage, larger and elongated. One of the subsets, the bottom one, still looked like monocytes, small and round. And one of the subsets, the middle one, looked like something in between. We used the Macroscreen platform to assess the function of these macrophages coming from different monocyte subsets, and we found great differences. As we already saw, the more elongated macrophages were less round. They also had the highest particle uptake and the lowest susceptibility to cell death. The in-between subset showed the highest inflammatory activity. This means that macrophages coming from different monocyte subsets have different functions. In Chapter 4, we cultured macrophages at different densities, so a different number of cells. Then we assessed their function with the Macroscreen platform. We saw that with increasing density, we also had a higher number of cells, meaning our setup worked. With increasing density, we also had an increasing particle uptake, but a decrease in lipid uptake and inflammatory activity. This means that the density cells were cultured at influences the function. In Chapter 5, we used 2 different growth factors called MCSF and GM-CSF and gave that to the monocytes to develop them into macrophages. At 4 different time points during their development, we used the Macroscreen to assess their function. This setup allowed us to compare the effects of MCSF versus GM-CSF, but also the functional changes over time. Comparing MCSF to GM-CSF, we found that in MCSF macrophages, particle uptake and lipid uptake were higher compared to the GM-CSF macrophages. Also, looking at the changes over time, we found that monocytes had a higher inflammatory activity and higher susceptibility to cell death compared to macrophages. To uncover patterns in the complex functional data, we used a computational method called principal component analysis. In this graph, every dot represents one time we did the Macroscreen experiments. And the closer 2 dots are, the more similar they are. To look at the underlying function mechanism, we also looked at the genetic changes. And if you compare the patterns in the two graphs, you see that they are similar. The distance between MCSF and GM-CSF is the greatest at the early time points at day 1, and the dots come closer to each other at the later time points. This means that MCSF and GM-CSF induce different programs for development very early on. We were also interested if MCSF and GM-CSF can be found in the plaque. For this, we used plaque from the carotid artery in the neck that was removed from patients during surgery. And then we measured the levels of MCSF and GM-CSF. We found that the level of MCSF was much higher than the level of GM-CSF. Also, when we divided the samples into stable plaque and ruptured plaque, we found that MCSF and GM-CSF were increased in the ruptured plaque. This means that in the plaque, macrophage function will be largely determined by MCSF, and the growth factors are increased in ruptured plaque. In Chapter 7, we used a different approach because there we used blood serum and measured how the macrophages react to it. So we didn't have only a single molecule, but a mixture of molecule in the serum. We also looked at a different disease, COVID-19, which emerged during my time as a PhD student. COVID-19 is a contagious respiratory disease that can have a range of functions, from mild cold to severe pneumonia. Interestingly, COVID-19 increases the risk to develop cardiovascular disease later in life, and macrophages in the lung are believed to play an important role in COVID-19. We used serum from COVID-19 patients that were so sick they had to go to the hospital, or serum from healthy people as a control and gave that to the macrophages. Then we used the macroscreen to assess the function of the macrophages. We found that in the macrophages who received the serum from the COVID-19 patients, particle uptake and lipid uptake were increased compared to the healthy people. While most of the COVID-19 patients were moderately sick and could be treated on the hospital ward, some of them got severely sick, and meaning they had trouble breathing or they had to go to the intensive care unit, the ICU. If we compared the moderately sick to the severely sick COVID patients, we found that particle uptake and lipid uptake were increased in the severely sick patients. Indeed, especially lipid uptake was associated with the clinical prognosis. In this graph, the area under the curve represents how well a function can predict if a patient will be severely sick. If you would flip a coin, the area under the curve would be 0.5. That corresponds to a random decision. You can see that the area under the curve for lipid uptake is larger, 0.77. This means that changes in lipid uptake can predict whether a patient will be severely sick. And all in all, the molecules in the serum change the macrophage function, and the disease environment influences their function. In conclusion, macrophage function can be changed at many stages during their development. This includes the monocytic origin or monocyte subset they come from, the density they were cultured at, growth factors they encounter in their development, and environmental challenges. In the future, our results could be used to develop therapies that change macrophage function during their development, to treat atherosclerosis or other diseases macrophages are involved in. Thank you for your attention, and I give the word back to the prorector.
Thank you very much for your presentation. The opposition will be opened by Professor Stoll, who was chair of the assessment committee and now secretary of the degree committee. And she is professor in genetic epidemiology and statistical genetics at the Maastricht University.
Thank you very much, dear candidate. thank you very much for a very clear presentation on a very complicated topic. As a geneticist, immune cells always scared me, and that's probably why I became a geneticist and not an immunologist. I would also like to congratulate your promoters for an apparently very successful PhD student. And I would like to start the opposition with a question which relates to your title of your thesis, Origin versus Context, which is, in fact, very interesting and a highly debated subject in the scientific community, as you also outline in your chapter, I think it was 2, on the review paper. So you discuss a lot about those classical versus intermediate versus non-classical monocytes, M1, 2, 3. And with increasing technology, single-cell sequencing, more and more monocyte subpopulations emerge. So how many subsets of monocytes do you expect, and how would they affect cardiovascular disease?
Highly esteemed opponent, thank you for your kind words and for your question. I think if you would really try to identify as many monocyte subpopulations as possible, you would be able to identify probably an immense amount of subpopulations. And I think it also has to do with what you define as a subpopulation, because as I also state in one of my propositions, the more markers you will include, the more subpopulations you will also identify. And I think in the future it will be very important to not only identify subpopulations, but also look at their function and see how they are distinct, if they are distinct, and also at steady state before you look at diseases, because I think it's very easy to look at a disease and see the monocyte subset, for example, distributions are shifting, and then say, oh, this subset is associated with disease, although it's still not clear how they act in steady state. So
may I ask you a specific question? If you go about the M2 monocytes, which are considered intermediate, do you consider them an intermediate transitory stage from M1 to M3? Or is it its own identity, origin, and is not considered an intermediate state?
Yeah, I think from the studies in mouse, but there's also a paper showing this in human, is that it's, they suggest a sequential development of the non-classicals from the classicals with the intermediates at an intermediate step. And I think from the results that we show in chapter 3, looking at the morphology, they really look like an intermediate state, because some of them have this elongated shape, some of them are really round. But looking at the function, they do have a distinct functional phenotype, which is really characterized by this high inflammatory activity, the high inflammasome activation. So I would be careful to saying they are just an intermediate state, because they also have a function that they exert.
So if we stay in that topic, so what's more important, the origin or the context? The genetics or the environment?
I would say, in the context, related to the macrophage, I think the context is more important, because strong polarization stimuli, for example, can really overrule any epigenetic or genetic imprints the monocytes have, and then they will respond more to the environment.
Okay, so that brings me to my next question. Chapter 5, you describe in your methodology how you stimulate the inflammasome on page 103. And you stimulate with you prime your monocytes with LPS. And then you wait for 3 more hours, and then you give your second stimulus to activate the monocyte for the inflammasome. What would happen if I would wait with the second stimulus a little longer and give the second stimulus a little bit later?
Thank you for your question. I am not sure what would happen, because I haven't performed this experiment in the lab. But if I would speculate, then I would say, comparing to macrophage that never got a first stimulus, you would see an increase in the inflammasome activity, but maybe less than a macrophage that received it within a shorter time frame.
Let me help you a bit. Where do you get LPS from?
Bacteria.
So, are you aware of a disease where bacteria causes systemic inflammation?
It could be in sepsis.
Exactly. So there's a large body of literature on if you pre-stimulate your monocytes with LPS and then wait for some time, you get a response like in sepsis, which is the opposite of the inflammasome because they get tolerized.
Okay.
And that could also be an explanation for the heterogeneity of your monocytes because some of them are sometimes pre-stimulated. But doesn't matter, it's super. Last question, innate versus adaptive immunity. In your chapter 5, you say you start with the innate immune response in your priming of your monocytes, and then the adaptive immunity comes in. Context versus origin. What's more important?
You mean for the...
For the development into the macrophages. I don't know either, you can just speculate.
I would say still the context, at least to the overbearing part, because in disease, the disease environment will be reflected systemically, and the monocytes that are recruited, this is a direct response to the depletion of tissue-resident macrophages and chemokines.
Thank you very much. I'm satisfied.
Thank you.
Back to the prorector.
Thank you, Professor Stoll. And the opposition will be continued by Dr. Westerterp from the Department of Pediatrics from the University in Groningen. Please go ahead.
Thank you very much, Mr. Prorector. Dear candidate, also I would like to congratulate you on your nice thesis, a very nice presentation, and I would also like to congratulate your team of promoters and co-promoters on that. And I would like to start with inviting your paranymph to read proposition number 2, one of your paranymphs.
Proposition number 2. Disregarding the effect of cell density on macrophage function introduces bias to in vitro experiments.
Okay, great. Thank you very much. And I think that proposition is mainly based on chapter number 4. And actually, I would like to start by asking you that if you think when the cell culture density is similar, we can compare monocyte-derived macrophages from 2 different individuals. Do you think that's the case?
Esteemed opponent, thank you for your kind words and for your question. I think if the density number is the same, you have more safe grounds to say that they are comparable.
Okay, and then, I mean, if we then look at figure 1, because there you do test that basically, you perform several assays to look into that. Are these correlations that we have here actually linear? So the more cell density, the more phagocytosis, are these linear correlations? Or?
I didn't test if they are linear correlations.
Okay, no, I was just wondering about that. Well, I have a particular interest in lipids, and so I was quite intrigued by your lipid uptake assay that you carry out in figure 1b. And can you perhaps elaborate a little bit more on which lipid you are actually looking at when you're performing this assay?
Yeah. So in the lipid uptake assay, it actually starts by isolating cholesterol from donor serum by density centrifugation, and then we oxidize the LDL fraction with copper sulfate and then dialyze it. So we will mostly look at ox-LDL, which we stain fluorescently using the body pie.
And which component of the ox-LDL is stained by the Bodipi?
I'm not sure exactly. I would need to inquire that with the manufacturer, but I assume it's maybe, it could be one of the protein parts of the ox-LDL.
Okay, yeah, cholesterol, phospholipids. I mean, maybe when we get a bit more of an idea when we look further at these pictures, because do you have any idea in which part of the cell the lipids would accumulate?
From the pictures we acquire with the HCA, it really looks like they are in the cytosol.
Okay, okay, great. Now I was just wondering, because also you just mentioned in your during your presentation that in the severely sick COVID-19 patients there were changes in lipid uptake, and I was wondering if you could, perhaps that was related to the cellular localization of the lipids. But okay, well then I would like to proceed to chapter number 6, which where you show that iron nanoparticles amplify macrophage foam cell apoptosis. And basically, you really focus on the apoptosis, but I was actually wondering if you have also looked into other pathways of cell death that could be mediated by these iron nanoparticles.
I think in this chapter we only focused on apoptosis.
And if you now think about that a bit more, because at some spots during the chapter you also look at some you look at inflammasome priming, you look at NLRP3, interleukin 1 beta. Of course, that's another pathway of cell death, but have you perhaps thought about some other pathways of cell death that could be induced by the iron ions?
I think it would be very interesting to perform the whole panel of functional assays that we can do with a macro screen also on these foam cell macrophages that have received the nanoparticles, because as we have seen that giving the antioxidants reduces this effect. So I would assume that performing a ROS assay, for example, would be very interesting, and that other functions related to inflammation will probably also be very interesting to look at.
Because did you look at apoptosis mainly an anti-inflammatory way of cell death? Did you also look further into that?
We didn't look further into that.
Okay. No, I was thinking, because it's iron ions, I was thinking that perhaps ferroptosis could be involved, because it seems to be so logical that it would be induced by iron. But
yeah,
it's maybe something for the future. And then I also would like to go with you to chapter number 5, where you look at the role of MCSF and GM-CSF in the monocyte to macrophage differentiation. And I would actually like to go with you to page 113 and figure 3D. And this connects a little bit to the question from the previous opponent, because do you think you're only looking at differentiation of monocytes into macrophages, or perhaps are these monocytes also differentiating into dendritic cells?
Yes, that could be possible, especially because GM-CSF is also known to differentiate the monocytes into dendritic cells. But we have performed an optimization experiment beforehand where we optimized the concentration of GM-CSF that we used. And I think compared to other concentrations found in the literature, it's now rather low, because indeed we saw that with a higher concentration, the monocytes took up a more dendritic cell-like shape.
Okay, so they did, because I mean it's interesting when you look at day 1 and day 3 that the MHC2 class protein complex is upregulated. There's a regulation of the adaptive immune response. It could be related perhaps to the dendritic cell-like phenotype.
Yeah, I think it could be related, but macrophages can also present antigens, so maybe it's also related to that.
Great. Well, thank you very much. I'm very satisfied with the responses, and I would like to give the word back to the pro-rector.
Thank you very much, Dr. Westerterp. Then the opposition will be continued by Dr. Wouters, who is a senior researcher in cardiometabolic inflammation here at the University of Maastricht.
Thank you. Dear candidate, also, I really enjoyed reading your thesis, and I especially enjoyed the review a lot, where you discussed really a lot of these pending issues in monocyte biology, and clearly identifying all the knowledge gaps that I also had. So that was comforting for me, but also shows that you are really on top of your game there. But obviously also the research quality, chapters was of high quality and sometimes a bit controversial even, so I liked it even more. And obviously that's also a reflection of the work of the promotion team, so congratulations for that. But before that, I would like to go on. I would like to give the other Paraninf perhaps the opportunity to shine and read proposition number 4.
Proposition number 4. The contribution of monocytic origin and growth factors to macrophage function challenge the current notion of local stimuli as a sole decisive factor to macrophage phenotype.
Thank you. It was already a little bit discussed in the first opponent, but I still have some questions on this, because if I look at chapter number 3, and you also showed the data just now in your beautiful presentation, actually only the macrophages from the classical monocytes look like macrophages and do what macrophages are supposed to do. So do you actually think that in our body this actually happens, that non-classical and intermediate monocytes turn into macrophages, and then is there any discussion on monocytic origin at all?
Esteemed opponent, thank you for your kind words and for your question. Indeed, actually, I feel very passionate about this because I have performed this experiment so many times, and every time the non-classical monocytes still look small and round like monocytes. But then you find publications in the literature that really state that all 3 macrophages, monocyte subsets, have the potential to develop into macrophages. And indeed, a very recent publication showed that a subset of interstitial macrophages in the lung is derived from the non-classical monocytes. So with the data I have gathered and what is known from literature, I believe that non-classicals can become macrophages, but they need some sort of additional stimulus that I do not have included in my experiments to become macrophages. Regarding this finding of the interstitial macrophages in the lung coming from the non-classicals, I would hypothesize that maybe GM-CSF is such a stimulus because it is the most prominent, most important growth factor in the lung for macrophage development. But I could also believe that it's maybe something related to erythrocytes, for example, or thrombocytes, because the non-classicals are believed to be the patrolling subset. So the question would here also be if they even leave the vasculature or they can be considered resident macrophages of the blood vessels.
That is really awesome, that answer, because I wanted to go back to chapter 2 on page 42 that you said that non-classical monocytes could be even considered as circulating macrophages. Then I made, in my notes, I can show them afterwards, the comparison to lung macrophages. So you kind of tackled that before I even asked that, so that's awesome. But what are the implications of that? So I always teach people there's no macrophages in blood, it's monocytes. Now you state that there might be, and then they're obviously stuck to the endothelium, right? So if we draw blood from a person, we get very little non-classical monocytes. Is that then because there are very few of them, or we just are not getting them out because they're stuck to the endothelium where they actually belong then?
Yeah. That is very interesting. I haven't thought about it like that, but I think it's definitely possible that they are somehow really in close contact with the endothelial cells lining the blood vessel. But at the same time, I think that it could also be that they are at a such low proportion of the cells or the monocytes in the blood because of the sequential development that I mentioned earlier. So if the classical monocytes are needed somewhere in the body where there's ongoing inflammation or injury and they are recruited to there, of course they are then in the tissue and won't develop into intermediates and non-classicals anymore. So I think it could also be that it's just the nature of this development that you don't find many in the blood.
Okay, something to be assessed later, I guess. And then I would like to perhaps briefly go to chapter 5 again, where you show indeed, you compare these two stimuli, CSF versus GM-CSF, and also literature already showed that it's probably more CSF than GM-CSF, and you tend to agree with that. But then looking at what we do see about GM-CSF in your data is when you project them on these plaque data, that only the day 1 signature pops up.
Yeah.
I found it interesting. So does that mean that there's a small surge of GM-CSF production very early on? And what does it actually mean? What kind of functions are there? I saw something on CD1, and then this might be lipid antigens being presented. So what are your thoughts on this day 1 signature popping up in the ruptured plaque?
If I'm not mistaken, I think you're referring to figure 6b, right? Where you see the signatures projected on the TSNE plot.
Exactly.
Yeah, and then indeed it is enriched in the GM-CSF signature, especially at day 1. And I believe that GM-CSF in the plaque is thus mainly responsible for the recruitment of the monocytes, but then for sustaining the population of monocyte-derived macrophages in the plaque, really MCSF takes over. So that maybe initially for the inflammation, GM-CSF attracts and recruits the cells, but then for sustaining the population, MCSF is more important.
Okay. Well, thank you. I'm sure we can continue for hours and hours, but I'm afraid I'm not allowed. So I'm happy with these answers, and I give the word back.
Thank you very much, Dr. Wouters. Then we will continue in line with a guest from Norway, Professor Akrust from the Rikshospitalet in Oslo. He's professor in internal medicine and in translational research. Professor Akrust, go ahead.
Cannot hear you.
Professor Akrust, we cannot hear you. Perhaps you have to unmute. Not yet. We cannot yet. Did you unmute already? Okay. So that's not the problem. Then there is some technical issue. Perhaps the technicians should have a look at it, and we do not wait. We continue with the next opponent, and we will come back to you. Is that okay? Good. Then we go to Professor Gerdes. He is from the Department of Cardiology of the Düsseldorf University. And his expertise is in cardiovascular immune biology. Welcome. Please go ahead.
Yeah, dear Mrs. Roeder, congratulations again for the presentation and putting together this very nice thesis. It was really a pleasure to read this, and also a pleasure to follow your presentation. I have a few questions about what monocytes and macrophages actually do in an organism that is not in disease. So what is the function actually there, and what do monocytes do when there's no inflammation?
Highly esteemed opponent, thank you for your question. I think at steady state, when there is no inflammation or injury that monocytes are urgently recruited at, they still circulate in the blood, and there might still be one or another macrophage somewhere in the body that needs to be replaced, because there's always also some cell turnover. So I think the monocytes will be involved in that, for example, and just keeping homeostasis in the body.
And do you have macrophages in other alternative sources of macrophages than those coming from the circulating monocytes?
Yes, you also have tissue-resident macrophages.
And what's the interplay of these tissue-resident macrophages with, how do they relate to macrophages and to the respective origin of the migrated or immigrated macrophages?
Yeah. So the tissue-resident macrophages are seeded in the embryonic development, so very early. They can also seed the fetal liver and then have in a second wave populate the tissues of the body. And in relation to the monocyte-derived macrophages, it is believed that, now with recent concept that there's this niche that the tissue-resident macrophages are embedded in. And when a niche becomes available, there are also an increase in trophic factors that will then attract monocyte-derived macrophages that will home in this niche and adapt the function of the tissue-resident macrophage that was in this niche beforehand. But it is also still under debate if this monocyte-derived macrophages that comes in to replace the tissue-resident macrophage will adopt exactly the same phenotype as the tissue-resident macrophage that was here beforehand, or if some of the functions might be different because it's an inflammatory monocyte-derived macrophage.
Okay, thank you. I would like to switch gears to something that monocytes and macrophages do with other cells. I mean, you're a lot focused on what macrophages or monocytes, and then with your Mac screen you do very nice and very standardized models of macrophage function, but you're not really looking at interaction of macrophages with or monocytic cells with other cell types. Mm-hmm. That's one thing I'm interested in is post-myocardial infarction wound healing and remodeling. What do you know about which monocyte subsets are involved in this process, not in the immediate inflammatory phase, but more maybe in the wound healing phase, and which are the main players actually in this interaction?
So from the literature review in chapter 5, we see that the intermediate monocyte subset shows an association with myocardial infarction. So I would believe that they are also involved in the processes after the myocardial infarction. But from what is known about the functions of the monocyte subsets, I would even speculate that the non-classical monocytes are also involved, because they are believed to have this wound healing, patrolling phenotype. And also one group has shown that non-classical monocytes preferentially develop into CD206 positive wound healing macrophages. So I would think that they are also involved.
What do wound healing macrophages do actually?
I would say they are involved in efferocytosis, so removing dead cells, for example, and also secreting anti-inflammatory macrophages to tone down the inflammation that was going on beforehand.
And do they affect other cell types that are then actively actually repairing the wound? And which cells would that be?
Yeah, definitely. I think they also react with other cells. In chapter 7, we show that the conditioned medium from macrophages, if you put that on the fibroblasts, this will increase the fibroblast count that we see. But that means that somehow via soluble mediators they communicate with the fibroblasts. So I could imagine that in a myocardial infarction, also cardiac fibroblasts could be encouraged by monocytes to produce collagen to repair the infarct.
Okay, good. And then one overarching question of your thesis in a way is that, okay, we have these 3 monocyte subsets that are circulating, but when we look at different tissues, and a lot of literature over the last years with single-cell sequencing and other novel techniques have identified so many different macrophage subsets, whatever. You read already proposition 6, that was actually what I wanted to ask, but you came beforehand. That is, we have a lot of markers, but what do we actually make out of this, and how can you speculate, or maybe it's a philosophical question, what is actually the definition of a cell type?
Yeah, thank you for your question. I think this really reflects how broad of a phenotypic plasticity these macrophages have. And one of the main messages of my thesis is also, I think, that you cannot only infer from one single stimulus that you have tried in vitro that this will be the function of a macrophage. It can be so much more diverse, and also not only the stimuli that a macrophage encounters when it's already a bona fide macrophage will influence the phenotype, because already during the development and at monocyte stage, it can be at least partly determined. So yeah, to come to the question if it's a cell type or not, I think is a very tricky one, especially in this context, because for some of the functional phenotypes that you see, I'm also wondering myself if this is maybe just a different activation state, or is it, like for the non-classical monocytes, is it just a more mature state compared to the classical ones? So yeah, I think this is really difficult because also there is not really consensus in the literature, and definitions of the subsets, for example, use of the nomenclature really makes it harder because it is not stick to very well. So I think also comparing results makes it more difficult to really determine, is this a separate subset? What are the functions of these subsets at steady state? And then look at the disease context.
Okay, thank you very much. I think it's good to hear that we're all struggling with the same problem. And thank you very much for your answers.
Thank you, Professor Gernes. And then we will go back to Professor Aukrust from Norway, and I hope that the technical issues are now resolved. Professor Aukrust, go ahead. Oké. Misschien even de technische dienst boven. Het werkt nog niet. Try again. The technical support is active, so you please speak again. Say something that. Oh.
Please continue with the next one. And for Professor Aukrust, please read your chat.
Okay, good. Then we go on, and we go to Dr. Silero Pastor, who is from the Maryland Institute and from the Maastricht University. Go ahead.
Thank you, Rector. So, dear candidate, first of all, I would like to congratulate you for this very nice thesis, also for a very clear presentation. Congratulations for that. And of course, to the promotion team, very nice work. And I would like to start my discussion with chapter number 3, page 62, figure number 3, where I do see some sort of donor variability in the data that you have shown us, interleukin-1 beta and interleukin-6. I do see some difference between donor B and C. They have an overall lower cytokine production. And in the thesis, you focus a lot on cell density, different cell populations. I'm just wondering about donor variability, because here you've measured only 3 donors. Mm-hmm. Can you elaborate on that?
Yeah. Esteemed opponent, thank you for your question. Indeed, also in chapter 5, we show that the density and the donor the monocytes are coming from will have an influence on their function. And I think this is also what is happening here in these results. I believe because it's different donors, it might be just interdonor variability that we see, or maybe differences in density, because what we observed in culture is that the non-classical monocytes and the intermediate monocytes seem to not adhere that well to the culture plate. At least we see that there are less cells in there, which is definitely a limitation of the findings in chapter 3. So I think that this is also where these differences arise from between the donors.
And can you think about maybe a more sensitive method to really look into this patient phenotyping? Because maybe you are looking actually at different patient phenotypes, perhaps. So can you think about a way of, looking more into detail of different cell populations per donor?
I think it would already be a good first step to ensure that the density is the same for all the 3 donors to exclude a bias that might be introduced in these results from this. And then also in the figure you are referring to, we are only showing 3 donors, but I think it would definitely be necessary to include more donors to make sure that these inter-donor variability is accounted for.
Okay, thank you for the answer. So you were mentioning right now the cell density aspect. If I look at, correct me if I'm wrong, but if I look at most of the figures in your thesis, you express the production of cytokines in picogram per milliliter. And you shown, I think, in supplementary figure 1 also the cell density, number of nuclei, also in different figures. And my question is, you haven't, or have you actually used the cell number or cell density numbers to correct for these concentrations or not?
Yeah. So one convenient thing with the macro screen is that when you perform the functional assays, you will receive the number of nuclei kind of as a bonus readout, because we use the host to stain the nuclei, and this is also what we focus on. So for the functional assays, you will always have the number of nuclei. And for example, in the apoptosis assay, we also sometimes use it to correct, because we see that there is no nucleus, but apoptotic signal. So maybe the nucleus is already disintegrated in this in apoptosis, so we use that to correct. But in the ELISA that we use, we don't directly get this cell number. So we we seed the cells at the same density when we plate the cells, but then after 7 days, of course, we can assume that the cell density might have changed, as we show in chapter 4, but unfortunately with the multiplex ELISA, we don't get it as this nice readout. But yeah, it would be nice to also have it, or maybe correct with just the amount of protein that is found in the well, and use that to correct the concentration or level of secreted cytokines.
But couldn't you use the medium, right, for the ELISA, let's say, while you could use perhaps with MTT or DAPI staining, then you have also the cell number. So actually from the same plate, you could actually have both things, and you could normalize, I think, in my opinion, in a better way, the ELISA values. Yeah,
that would definitely be an option to correct for it. Yeah.
Okay, yeah. Thank you for the answer. Then this microscreen platform, you have used it, but actually it is the first time that I see this platform. I'm very curious to see how it works. Do you get absolute values? Do you get relative numbers? Can you maybe explain me how it works more in detail?
Yeah. So the Macroscreen platform is based on the BD pathway, which is an automated fluorescent microscope. And for all the assays that we perform are based on fluorescence. Yeah, they are fluorescence-based functional assays. And then what we receive from the Macroscreen is an image that is composed of 9 images in total to make up a big one. And then to analyze this image, we first segment on the nuclei to identify the cells. And then we assume that an enlarged nucleus will encompass the cell. And then we measure the fluorescent intensity within this region that we specified as a cell.
Okay, so what about sorry that I interrupt you. So what about the application of microscreen in a clinical setup? Because you mentioned that in the impact chapter, if I'm correct, you also discussed the possibility of using it more like in a clinical setup, but you don't have absolute numbers. Is that correct? You get like a relative abundance. So can you elaborate on that? How would you envision that if you don't have absolute numbers or concentrations, let's say?
Yeah, I think as the HCA or Macroscreen as it is at the moment is not fit yet to be employed for really patient care. But how it can be implicated in patient care is using the knowledge that we get from the Macroscreen about the macrophage function and then use this functional phenotype in a disease to think of treatments. For example, in COVID-19, we see an increased phagocytosis. So with this knowledge, we can think of a way to maybe target phagocytosis if we see that it is not beneficial in the disease. So I think it's not so much about actually using the macro screen to treat, as treatment option or like a diagnostic option, but more about generating knowledge that can be used as a basis to think of therapies that target macrophages.
Towards personalized medicine, perhaps.
Yeah.
Okay. Thank you. Very clear. Then I would like to move to chapter 4, if we still have time. Yes. You start the chapter with this concept of quorum sensing, right? But as you know, of course, quorum sensing is related to this bacterial metabolic exchange. And I'm just wondering why you don't really mention any metabolite at all. So you go back to the concept of different cell densities, having different functions, but you don't really explore actually whether there might be a different rate of metabolic exchange depending on the cell density.
Yeah. I think that would be very interesting to do, actually. You're right, we're not doing it right now, but it would be very interesting to complement this chapter with some metabolic data.
Okay, and any idea what kind of metabolites you would like to target? What kind of pathways?
I think I would first look at the maybe more obvious ones, like glycolysis, for example, because I can imagine that also if there's less cells in a well, then more of it will be available, if that also changes how it is used by the cells.
Okay, thank you very much for the answer. Still look at the prorector. Then we continue. We go now to chapter number 5, lipid uptake. It has been discussed before. Again, you mentioned that you focus mostly on cholesterol and oxidation of LDL. What's the reason of focusing on these lipids? Because, of course, there are so many lipids. Can you elaborate on that, perhaps?
Yeah. So we chose ox-LDL because it is implicated in atherosclerosis. With the oxygen radicals that are found in the lesions, of course, the LDL will be modified, so oxidized. And I think this was the main reason that we used the ox-LDL. But in chapter 6, in fact, we also used the VLDL to generate foam cell macrophages to which we then give the iron oxide nanoparticles.
And wouldn't you analyze as well phospholipids, because they're an important class of lipids that are actually part of the membrane, right? So if you're talking about uptake, I can imagine that the composition of the cellular membrane in terms of phospholipid content is also relevant.
Yeah, we don't look at the composition of the membrane, but I think it would be very important to look at it. And also with this lipid uptake, we could, or related to lipid, we could even look at another function, which would be cholesterol efflux. We don't have an assay that accounts for it yet, but I think especially in the context of atherosclerosis, it would be very interesting to look at that, because it is not only the uptake of lipids that makes a foam cell, but also if the efflux is impaired, that these LDL accumulates in the cell and generates the foam cell. I was finished.
Well, okay, great. Thank you. Adele Ruder, the time appointed for defending your thesis has passed. The degree committee will now withdraw to discuss the quality of your thesis and your defense. I request that you and your company await the results of our deliberations and our return in this room.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
10 miles in my rearview mirror, I know what it felt like. My goals only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. On the line, I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goals only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Make the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. And even though these nights seem long, I know that I am never on my own. Know you've won the battle once you've grown. South side, 10 miles in my rearview mirror, I know what it felt like. My goals only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goals only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile.
Adele Veronika, the degree committee here present has assessed the quality of your thesis and your defense. In view of its positive verdicts and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor Biese is authorized to confer upon you this academic distinction in accordance with Dutch university custom. I invite your supervisor now to take the floor.
Dear Dr. Roeder, before I grant you the thesis, I have to ask you a serious question. Do you promise to work in accordance with the principles of scientific integrity at all times? To be careful and honest, transparent, independent, and responsible?
Yes, I promise.
Okay, good. By the authority vested in us by law, in conformity with the decision of the committee here present, I hereby confer upon you, Adele Veronika Roeder, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary, and the other members of the committee, and affixed with the official seal of the university. Now, hopefully it works out. This time, I've been practicing a lot on this, but...
Dr. Ruder. Dear Adele, what a great honor it is to be the first person to address you in this way and to congratulate you with this extraordinary achievement. Natürlich möchte ich auch deine Eltern, Matthias, und dem Rest deiner Familie meine Glückwünsche aussprechen. I am ashamed to say my knowledge of German is limited at best, so I will have to continue the rest of my speech in English. Adele. Erik and I sometimes refer to you as the queen of metaphors. As everybody here has witnessed, you are exceptionally talented in finding the right image to clearly transfer a complicated message in the most natural and seemingly easy way. We can be a tad less organized or clear in our communications. I will therefore shamelessly make use of your metaphor and claim that the PhD trajectory can also be considered a race, with twists and turns, friends running alongside you, coaches falling asleep, unforeseen obstacles, and environmental conditions affecting your run. You started 5.5 years ago with a warm-up run, an email with the subject, Inquiry about Internship Possibilities. I quote, I was excited to find Karim. I feel a strong desire to learn even more about biomedical processes in our body and contribute to ongoing research. I think we can all agree that you have achieved that. So, in the spring of 2018, you joined me, Margot, and Suzanne and started high-content imaging of macrophages. After a couple of weeks, Erik and I already agreed that you would be a great PhD candidate. Judith became part of your supervisory team too, and that was that. You were part of the PhD race. Bold and brave choice, if you ask me, as you had been given the whole tour of the hospital by our lab technician Matrouche, which can be a bit daunting. You started out as a quiet runner, a bit hesitant at first maybe, but with very clear goals in mind. Your drive and motivation were there from the start, and more you took control of your own path. You decided to take an extra course in statistics, you became more and more confident in project management, etc. Close to the finish line, you even made a Gantt chart for all of your supervisors and co-authors, giving us all very specific to-dos and deadlines. Every 2 weeks, the chart was pulled out again, and we received red boxes when we were not doing our duties. I have a feeling we have not seen the last of this system. And now I have to fix my own system. Okay. Looking back, you achieved many milestones in your race. You quickly became a cell profiler expert, and your pipelines have become standard in the lab. You noticed culture density is a critical factor in macrophage biology and turned this into a really nice publication. You presented your work at conferences and received the highest tutor feedback marks in the history of our lab. 384 well plates turned out to be just a walk in the park for you, as well as designing and analyzing huge imaging screens. You are leaving behind a wealth of data, truly valuable for years to come. Of course, your race also contained obstacles. The one stone in your shoe that kept coming back was the human monocyte project. These cells, they just refused to be sorted and sequenced by you, but you did not give up and still managed to study their behavior. The Frontiers in Immunology review process. This appeared sometimes more to be a copy of the never-ending story, which tested even your patience, and that is saying something. And then there was COVID, the big environmental disturbance of your race. And suddenly, your apartment table had to become the center of your universe, the place where you work, eat, meet friends online in meetings, and relax. That's not easy for anyone. And thankfully, we could go back to the lab after the first couple of months. All along your run, you've had friends and family cheering you on. Colleagues running alongside you became good friends. Chang, Jan, Olivia, René, Valeria, Elias, many others. But you also cheered on and helped everyone around you. You are responsible, gentle, helpful, with a delicate sense of humor and an eye for everybody in the lab. You made the most beautifully handcrafted cards and Secret Santa surprises, and very tasty home-baked goodies. Your experimental work and data are extremely reliable and trustworthy. Seeing Adele on lab bottles has now officially been recognized by the university as a quality mark. Moreover, you became an I'm Karim PhD representative and co-authored a guide for PhD students, resulting in an award during the Karim Annual Symposium, which you are clearly thrilled to receive, or not. Overall, yes, a PhD is like a race, and you have run it very, very well. Achieved what you set out to do, expand the body of science, and defend it in front of a jury of experts and peers. But in my view, a PhD is so much more than just a contribution to research. You become a doctor of philosophy. Philosophy, meaning love of wisdom. It is a journey during which you grow, not only as a scientist, but perhaps even more so as a person. You open yourself up to learn, you find your way in life. And it has been my great privilege to see you do just that. And I think in the coming months, you will find your way through the US. So I wish you all the best doing that.
Thank you.
Dear Dr. Rüder, also on behalf of the Maastricht University, I congratulate you with the degree you have acquired and also with the very scientific, solid and highly informative work you have done. I think it's still for you not sure whether you want to continue your research, so you took a sabbatical and traveled a lot, but your decision has not yet been made? Okay, good. So I wish you a lot of luck in your future career and also in your personal life. I'd like to thank the opponents from outside Maastricht, Dr. Westerterp from Groningen, Professor Aukrust from Oslo. It's a bit of a pity that the technical issues couldn't be solved, so you were not able to ask the questions you had. We apologize for that. Thank you, Dr. Gerdes, coming from Düsseldorf to Maastricht. And then we come to the end of this academic session, and we have just a practical issue. We will take a photograph with the team here in the Corona and Professor Aukrust, and afterwards we will take a photograph on the stairs. So the public, your friends and family are allowed to leave the room now and to go already to the Refter, where we will join you in a couple of minutes. So then we close this academic session and we wish you all the best for the future. Thank you.